Hello, we are group 3.2 and we are going to be explaining how to use genetic engineering through mechanisms like homologous recombination and then procedures like PCR and electrophoresis to remove the IRA2 gene from yeast. So, for homologous recombination, this is how DNA recovers from a double-strand break. So this is a strand of DNA. Double-strand break is when it tears right down the middle through both strands. The first response of the cell when the DNA experiences a double-strand break is for enzymes to come in and chew away at one of the strands on each of the strands. It then locates a piece of homologous DNA. The strand that the DNA finds is homologous to it. It can bind one of the strands to one of the homologous DNA strands after it is separated, like so. Then that end of the DNA acts as a primer which attracts DNA polymerase. DNA polymerase can then start transcribing in this direction using the homologous DNA as a template and forming a long strand of our DNA. After it's over, we end up with a strand of our DNA, and the template DNA rebinds to itself since it was just a template. Then this new strand comes up and can bind, since it is homologous, to the bottom strand of the DNA where the double-strand break occurred, therefore fixing the double-strand break. We are going to use this mechanism of homologous recombination to try to genetically engineer yeast to remove our gene IRA2. And I'm going to tell you a little bit about polymerase chain reaction, or PCR. This is a laboratory procedure that's used to amplify a short piece of DNA into millions and billions of copies to then use in research. So the reason that we are using PCR in this lab is to amplify a short piece of DNA with our gene in it, IRA2, so that we have lots of DNA with lots of IRA2 genes in it, and then we can use that for our next steps. In polymerase chain reaction, an existing strand of DNA is used as a template, and primers and DNA polymerase are added in order to copy the primers in just the area of the DNA that you want.
To do PCR in our lab, what we did was we first created primers of the genome of our yeast using the genome database and located our area of interest, which is IRA2. Then we found the primers 500 base pairs back and 500 base pairs forward, which is the forward and reverse primers. And we combined water, a buffer, DNTPs, which is nucleotides, primers, template DNA, and DNA polymerase in a vial to create a 50 microliter solution. We would then put the vial in a centrifuge to just make sure it's well mixed, and we put the vial in a thermal cycler where it will perform PCR.
Cool.
In the thermal cycler, the DNA is taken through a couple of steps in order to perform PCR. First, the DNA is denatured, the temperature is raised to 98 degrees Celsius, and the DNA separates like so. The next step is annealing. This is when the DNA is taken through a couple temperature cycles. First, it's cooled down so that the primers can attach there and there. Next step is extension, in which DNA polymerase binds to the primer and starts transcribing DNA on both of the strands. This creates two strands of DNA with the primers and with the transcribed DNA. This whole process is then repeated many times to create many strands of your segment of DNA, and then there is a final hold at 4 to 10 degrees Celsius to renature the DNA and finalize the process.
In order to see if our PCR work, we could use a process called gel electrophoresis, which allows us to visualize the DNA strands, which is the results of PCR, by separating the strands by size and charge. This is done by pushing the molecules through an electric field in a gel, and since DNA contains a small charge, it would be attracted to the opposite charge and it would be pushed, and depending on the size of the DNA, it would be pushed a certain length. And then a DNA ladder would be used to determine how big the size of the DNA is and if your PCR result was a success.